Greetings. Welcome back to 0610 Explain. Today we are going to do paper number 6, Alternative to Practical, October November 21, code number 61. So, this is a paper where we are going to start reading the question. It is a dialysis tubing that can act as a partially permeable membrane because it has microscopic holes. So, let me first explain what do I understand by the term partially permeable membrane. This is a diagram which is presented over here to show you that these pores that you can see here on the screen, these are tiny pores. Now, as we can understand, there are two types of molecules or particles over here. This is the blue ones and the red ones. As you can see that the blue ones can easily pass through this gap, but the red one cannot. So, I can say that this partially permeable membrane will allow the blue particles, that is the water molecules, to pass through, but it cannot let the sucrose, that is the red molecules, to pass through. So I am saying that this partially permeable membrane is permeable to water but not permeable to sucrose. Coming back to the question paper. The student investigated the molecule that can diffuse through the dialysis tubing. I just explained what do you mean by the term. The piece of dialysis tubing was soaked in water and a knot was tied at one end. So you can see this test tube over here, the diagram which is presented. So they have put one dialysis tubing over here. In this they have added 20 cm cube of a solution. Outside you can see that they have knotted it on one end. On the outside of this tubing you can see they have added distilled water. On the other side of the tube they have hung it on the side and they have put an elastic band so that it will not slip or move out. And they have a solution M inside is a dialysis tubing bag. Okay, so this is the conditions provided. The unknotted end of the dialysis tubing was opened and 20 cm cube of solution M was put into the dialysis tubing. Now, the outside of the tubing was rinsed. So this is very important so that there is no impurities or contamination and so that it will not affect your final result. The dialysis tubing bag was put inside a large test tube and the open end of the tubing was folded over the top of the large test tube. I have explained all this. The large test tube was then filled with distilled water. After 15 minutes, the dialysis tubing was removed. So they have kept this in the this condition for 15 minutes, and after 15 minutes, the dialysis tubing inside and the water outside both are going to be tested. So here the outside was the beaker which was put as labeled T. 2 cm cube of the samples of liquids were tested for reducing sugar, protein and starch. So as we do a quick revision, reducing sugar, the reagent that we are looking at is Benedict's reagent. Then we are talking about the protein where we are talking about biuret, and the starch is the iodine solution. You can have a look at these particular color modules that I have kept ready for you. So here you can see that this is a dialysis tubing original color. Sorry, this is a Benedict's reagent original color, which is blue color. As the amount of sugar increases, it will move from green, yellow, orange to brick red, which confirms the positive results. Then we are looking at the biuret test over here. So the biuret test, you can see the original color is blue, and when the color changes or when the protein is present, it is turned to purple, lilac, mauve. All these three colors or names are allowed in the board. Then we are looking at the iodine reagent over here. So iodine, as you can see, this is the original color, which is yellow brown, and once the color changes, it becomes blue black. These are the colors that are used for reference. So coming back to the paper, the student results are shown in the figure 1.2. Solution M, the colors are mentioned. Solution T, the colors are mentioned. They are saying prepare the table to record the results shown in figure 1.2. So we are going to quickly have a look at how the table will look like. So what the observations are: the dependent variable. The dependent variable will always go on the right side of the table and the independent variable will go on the left side of the table. Since we are going to have three observations regarding the reducing sugar, proteins and the starch. So this is how you are going to start making the table. This is the solution section. Color change observed. Then we are looking at the reducing sugar color. Then there is protein. And the last one we are writing is starch. There were two solutions which were looked at. There was solution M and T. As we will quickly see the colors, there was orange and orange in both. Then here we are talking about purple and blue in the proteins. And the last one, starch, blue, black and brown. So this is a paper which is going to be, this is the table which is going to be marked for 3 marks. State the substances or the names of the food test. We have already spoken. Reducing sugar is Benedict's reagent, protein is biuret and the starch is iodine. State which substances are present in solution M. So as we see in the solution M, the color has changed to orange. That is the first stage of here. We are looking at the orange color. That means that moderate amount of sugar is present. Then we are looking at the proteins. So in proteins, we said that the purple color is seen in M. So it contains proteins and it is also showing blue-black color of the starch. So all these three are present over here. So starch, protein, reducing sugar. Since the question is for 1 mark, you need not write anything more than that. Conclude based on the results if any of the substances in solution M diffused through the dialysis tubing. So as we see in the diagram that in the dialysis tubing there was something and then outside there was water. But when we tested solution T from that water, it did show you orange color. That means that the reducing sugars have diffused. So we are looking at the substance reducing sugar. Evidence is that the positive test orange for substance was seen. Explain why it is important to rinse the outside of the dialysis tubing bag in step 3. Some of the solution M might have got into the outside of the tubing or to avoid any type of contamination. Identify one hazard for this investigation. So as we know, these food testing reagents are corrosive. So you need to wear gloves or eye protection or goggles. There can be one more hazard where we are looking at that we are going to use the Benedict's test. In Benedict's test, as we know that the procedure is that you heat it in a water bath at 80 degrees Celsius for 2 to 3 minutes. Okay, so during this time there can be a hazard of hurting yourself. The water is warm, etc. So the precautions that you can talk about is stand up during practical work, use of water bath, use of gloves or use of tongs. Moving ahead, we are looking at the starch can be broken down into reducing sugar. An enzyme amylase catalyzes the reaction. So, in this particular question, it is very important to see the marking points. It's a 6 marks question. The format of the answer is already written for you. As I'm just going to explain what are the main marking points over here. So the first marking point is that when you are going to use the effect of pH. So for the pH effect, we are going to see two different test tubes labeled A as with pH 2, that is acidic pH, and test tube B with pH 8, which is going to be a basic pH. Okay. So this is going to be a first mark. Second mark we are looking at is the constant variables, which will be for 2 marks rather. So the 2 marks which you can grab is for concentration of the amylase. So how are you going to make the starch solution first? So you're using 20 grams of potato and adding 10 ml of water, crushing it with a mortar and pestle. Then you're going to keep the same starting temperature. How you're going to maintain the temperature using a thermostatically controlled water bath. Adding 10 cm cube of amylase, all this can be your second mark, 2 marks. So 2 plus 1, so that's like second and the third mark. Okay, then we are moving ahead. Using of an appropriate reagent. So here we are going to use what is called as the iodine solution. So iodine solution name is going to carry the third mark. So this is your third mark. Then we are going to use a spotting tile to observe the color change or place it on a white tile. So this white tile is going to give you your next mark. Okay, then we are going to also make use of the observations or repeat the experiment. So this is your fourth mark. Then we are going to repeat the experiment using the same conditions. This is your fifth mark. Okay, and we are going to look at the safety precautions for your sixth mark. Okay, so these are the 6 marks, how it is allocated or how it is how the marks are gained in this question. Okay, if you like the explanation and the way I continue doing the questions, please do like and subscribe. Moving ahead to the next question. This is a new question. The earlier one was for 17 marks. Moving ahead to another 23 marks of the question, because this is a 40 marks paper. The student used an aquatic plant to investigate the effect of temperature on the rate of photosynthesis. So this is an experiment which is done. The lamp is kept at a distance which is showed as 10 cm3. The further the lamp will move away, the temperature will be reduced. The closer you bring the lamp, there will be more intensity of the light and more temperature, or the temperature can be increased. Okay, so here we are looking at state the variable that was changed. So it is the temperature. State two variables that should be kept constant. Now there are many variables that can be kept constant. So we are looking at the light intensity or distance of the lamp, color or wavelength of the light, timing for collecting of gas, as in you're looking at 2 minutes, 5 minutes, whatever. Type or species of the or mass or age of the plant. Okay, this is again very important. You are using a plant over here. It should have the same number of leaves and all. Amount, mass or concentration of hydrogen carbonate or the carbon dioxide added. Okay, so all these are the important constant variables which you need to monitor during the experiment. Only one set of result was collected. Explain why repeating the investigation 2 or more times would be important in this method. Your answer would be to identify any anomalies. Okay, anomalous results, something which is not falling in place. Sometimes there is a condition where you have taken a wrong reading. So this is called as anomalous results. Now there is a table which is giving you the readings of the volume of gas collected and the rate of photosynthesis. Calculate the percentage increase. So percentage increase formula is final minus initial upon initial into 100. So here they are asking you produce from 10 to 15. So here we are going to look at 10 was 7.8, 15 is 12. So 12 minus 7.8 upon 12 into 100, your answer is going to be 53%. That's for 3 marks again. Use the information in the table 2.1 to plot the line on the grid to show the effect of temperature on the rate of photosynthesis. Here it is very important that you label the axis. So your x-axis and y-axis with the correct labeling is going to fetch you 2 marks. Then the plotting of the points is going to fetch another mark. Again, I have mentioned in the earlier videos that you need to do more than 60 or 70% of the graph space available. So that is what I have used properly. You could also write the scale in this edge, and now your work is to just join the dots. After plotting them, always try to make a smooth curve. Do not try to break the edge. Then that is continued. That is considered that your line is broken and your marks will be cut. So again, this diagram, this graph is for 4 marks. Describe the pattern shown by the data in the graph. So as the temperature increases from 0 to 20, we can see 20 here. The rate of gas collected increases from 0.25 that you can see here to 1.14 as you can move ahead here. Use the graph to estimate the rate of photosynthesis when the temperature is 17 degrees Celsius. Show on your graph where you took your reading. When they are mentioning things like this, it's a 2 marks question. 1 mark will be allocated for the marking on the graph and 1 mark for writing in this dotted line. So 17 degrees Celsius would be somewhere here. If I take this line forward, and wherever it is meeting this graph, and I'm going to move this line here like this, so it is going to be somewhere around 0.9. Okay. Now there is a diagram which has to be provided, and they have asked you make a large drawing of the flower shown and label the stigma on the drawing. Now when you are looking at these kind of drawings, sometimes it looks very difficult for those students who are not very good at drawing. Please be rest assured they are only asking you to make 7 petals. 1, 2, 3, 4, 5, 6, 7. Okay, so we are talking about 7 petals, 5 anthers, and we are looking at the diagram or the flower should look similar to what they have drawn, and they are asking you to draw the stigma. I have kept the diagram of a proper flower ready for you all. So you can see that this is the stigma. We are seeing there is this stigma, anther, and the filament, which is called as the stamen or the male reproductive part. The sepal protects the flower in the bud condition. Then we are looking at the stigma, style, and the ovary and the ovule over here, and the stalk. This part is called as the stalk. Okay, so this is the typical flower. So here when you need to label the stigma, so now we know the stigma is the center part here. Okay. 5 marks for this diagram. If you just make the flower look like it, you are going to get all your marks. Next question, we are looking at the length of line XY. So if you are looking on the diagram, they have already shown you X and Y labeling. You just need to do the drawing or the measurements. Always remember, I repeat in my videos that measurements will always be in millimeters. So according to me, this should be around 90 millimeters. The magnification formula is already provided to you. You need to find the actual length. Now here always read the question properly. Calculate the actual width of the flower shown using the measurement and the formula. Include the unit. Generally, they will always write the unit here at the edge. But in this question, they haven't written, and they are expecting you to write, for which they are giving you 1 mark. Okay, so you need to be very cautious as to what they are asking. So this is their magnification was 6 times. How did we come to know about that? Here it is mentioned that it is the magnification here is 6 times. The length of X and Y we have already taken as 90. So 90 divided by 6 will be 15. So that is your answer, 15 millimeters. 1 mark for measuring 90, 1 mark for doing the proper calculations and writing 15, and the last mark was, the third mark was for writing the millimeters. Okay. So this was the paper 61. I hope you like the paper explanation. Do like, subscribe, ring the bell icon, and wait for the next video. Thank you.